Red blood cells might have a lot of changes, but is there a relationship between those changes and different blood type group? Is this change is related to the change of the blood type group? Is this change is related to change in blood type group or a change in the bone marrow? Does this change in blood type group affect your bone marrow or even the bone itself? And if we take in consider that this change will affect the bone, so it will affect the shape of your body. Question: is the structure of red blood cells is different in different red blood cell type? I mean, in other words, are the morphologic changes in red blood cells have a relationship with blood type group? Question: does this function is affected by the change in blood type group, the function of red blood cells? To the production of red blood cells. Human erythrocytes are produced through a process called erythropoiesis, developing from committed stem cells to mature erythrocytes in about 7 days. When matured, these cells circulate in the blood for about 100 to 120 days, performing their normal function of molecules transport. At the end of their lifespan, they degrade and are removed from circulation. Erythropoiesis is the process in which new erythrocytes are produced, which takes about 7 days. Erythrocytes are continuously produced in red bone marrow of large bones at a rate of about 2 million cells per second in a healthy adult. Erythrocytes differentiate from erythropoietic bone marrow cells, a type of hemopoietic stem cells found in bone marrow. Unlike mature red blood cells, bone marrow cells contain a nucleus. In the embryo, the liver is the main site of red blood cells production and bears similar types of stem cells. At this stage of development, thus this process is affected by the change in blood type group. Erythropoiesis can be stimulated by the hormone erythropoietin, which is sensitized by the kidney in response to hypoxia, systemic oxygen deficiency. In the last stages of development, the immature red blood cells absorb iron, vitamin B12, and folic acid. These dietary nutrients that are necessary for proper synthesis of hemoglobin, iron, and normal red blood cells development, B12 and folic acid. I mentioned a question about the might relationship between iron, B12, and folic acid. Is there might relationship between a blood type group and erythropoietin? We know that the absorption of iron, B12, and folic acid may be different from a person to another due to different diseases or aspects which may be related to blood type group. On the other hand, thus the concentration of iron, B12, and folic acid is different in blood type group due to that deficiency of any of these nutrients may cause anemia, a condition in which there aren't enough fully functional red blood cells carrying oxygen in the bloodstream. Just before and after leaving the bone marrow, the developing cells are known as reticulocytes. These immature red blood cells that have shed their nuclei following initial differentiation after 24 hours in the bloodstream, reticulocytes mature into functional red blood cells A lot of processes might have a relationship with blood type group. In traditional medicine, they told us that the manufacture of red blood cells may be affected by a lot of aspects, maybe some foods. On the other hand, those foods may be harmful to the change in blood type group. Apoptosis, a form of apoptosis, programmed cell death, is the aging and death of mature red blood cells. As a red blood cells ages, it undergoes changes in its plasma membrane that makes it susceptible to selective recognition by macrophages and subsequent phagocytosis in the reticuloendothelial system, spleen, liver, and bone marrow. Thus, this change in plasma membrane is related different in different blood type groups. Thus, this process is affected by changes in food, which may affect liver, spleen, or even bone marrow. This process removes old and defective cells and continually purges the blood. Apoptosis normally occurs at the same rate of apoptosis, keeping the total circulating red blood cell count in a state of equilibrium. Many diseases that involve damage to red blood cells, hemolytic anemia, sepsis, malaria, pertussis, or nutritional anemias, or normal cellular processes that cause cellular damage, oxidative stress, may increase the rate of apoptosis. But we all know that malaria may happen in some blood type groups more than others. Even nutritional anemia may be different between different blood type groups. Conversely, erythropoietin and nitric oxide, a vasodilator, will inhibit apoptosis. Following apoptosis, the hemoglobin content within the red blood cells is broken down and recirculated throughout the body. The heme components of hemoglobin are broken down into iron ions and a green bile pigment called biliverdin. The biliverdin is reduced to the yellow bile pigment bilirubin, which is released into the plasma and reticulated to the liver, then bound to albumin and stored in the gallbladder. The bilirubin is excreted through the digestive system in the form of bile, while some of the iron is released into the plasma to be recirculated back into the bone marrow by a carrier protein called transferrin. This iron is then reused for erythropoiesis, but additional dietary iron is needed to support healthy red blood cells life cycle. I asked a question about the might relationship between a blood type group and its inside, not only outside structure, which might be affected by a lot of factors or disease which are related to blood type group. Capacity of the bone marrow to produce red cells is enormous, which when stimulated to peak activity and when provided adequately with nutrient substances, the marrow can compensate for the loss of several pints of blood per week. Hemorrhage or accelerated destruction of red cells leads to enhanced marrow activity. The marrow can increase its production of red cells up to 8 times the usual rate. After that, if blood loss continues, anemia develops. The rate of erythropoiesis is sensitive to the oxygen tension of the arterial blood. When oxygen tension falls, more red cells are produced and the red cell count rises. For this reason, persons who live at high altitude have higher red cell counts than those who live at sea level. For example, there is a small but significant difference between average red cell count of persons living in New York City at sea level pressure and persons living in Denver, Colo, more than 1.5 kilometer, 1 mile above sea level. Where the atmospheric pressure is lower, natives of the Andes living nearly 5 kilometers, nearly 3 miles above sea level, have extremely high red cell counts. The rate of production of erythrocytes is controlled by the hormone erythropoietin, which is produced largely in the kidneys. When the number of circulating red cells decreases or when the oxygen transported by the blood diminishes, an undefined sensor detects that change and production of erythropoietin is increased. The substance is then transported through the plasma to the bone marrow, where it accelerates the production of red cells. The erythropoietin mechanism operates like a thermostat, increasing and decreasing the rate of red blood cells production and accordance with need. When a person who has lived at high altitude moves to a sea level environment, production of erythropoietin is suppressed. The rate of red blood cells production declines and the red cell count falls until the normal sea level values is achieved with the loss of 1 pint of blood. The erythropoietin mechanism is activated, red cell production is enhanced, and within a few weeks, the number of circulating red cells has been restored to the normal value. The precision of control is extraordinary, so that the number of new red cells produced accurately compensates for the number of cells lost or destroyed. Erythropoietin has been produced in vitro outside the body by the technique of genetic engineering, recombinant DNA. The purified recombinant hormone has promise for persons with chronic renal failure who develop anemia because of a lack of erythropoietin. Survival of the red blood cells in the circulation depends upon the continuous utilization of glucose for the production of energy. Two chemical pathways are employed, and both are essential for the normal life of the red blood cells. An extraordinary numbers of enzyme system participates in this reaction and direct the energy evolved into appropriate uses. Red cells contain neither a nucleus nor RNA ribonucleic acid necessary for protein synthesis, so that cell division, mitosis, and production of new protein are impossible. Energy is not necessary for oxygen and carbon dioxide transport, which depends principally on the properties of hemoglobin. Energy, however, is needed for another reason, because of the tendency for extracellular sodium to leak into the red cell and for potassium to leak out. Energy is required to operate a pumping mechanism machine in the red cell's membrane to maintain the normal gradients. Differences in concentration of these ions. Energy is also required to convert methemoglobin to oxyhemoglobin and to prevent the oxidation of another constituents of the red blood cells. Cells have an average lifespan of 120 days. It is different in different blood type groups because red cells can't sensitize protein reparative processes are not possible as red cells age. Wear and tear leads to loss of some of their protein and the activity of some of their essential enzymes decreases. Chemical reactions necessary for the survival of the cells are consequently impaired. As a result, water passes into the aging red cells, transforming its usual discoid shape into a sphere. These spherocytes are inelastic, and as they sluggishly move through the circulation, they are engulfed by phagocytes. Phagocytic cells form a part of the lining of blood cells, particularly in the spleen, liver, and bone marrow. These cells, called macrophages, are constituents of the reticuloendothelial system and are found in the lymph nodes, in the intestinal tract, as free, wandering, and fixed cells. As a group, they have the ability to ingest not only other cells but also many other microscopic particles, including certain dyes and colloids. Within the reticuloendothelial cells, erythrocytes are rapidly destroyed. Protein, we all know the different blood type groups need different quantities, qualities, and types of protein, including that of hemoglobin, which is broken down, and the component amino acids, which are transported through the plasma to be used in the synthesis of new proteins. The iron removed from hemoglobin passes back into the plasma and is transported to the bone marrow, where it may be used in the synthesis of hemoglobin in newly forming red blood cells. Iron, not necessary for this purpose, is stored within reticuloendothelial cells but is available for release and reuse whenever it is required. In the breakdown of red cells, there is no loss to the body of either protein or iron, virtually all of which is conserved and reused. In contrast, the porphyrin ring structure of hemoglobin, to which iron was attached, undergoes a chemical change that enables its excretion from the body. This reaction converts porphyrin, a red pigment, into bilirubin, a yellow pigment. Bilirubin released from reticuloendothelial cells after the destruction of erythrocytes is conveyed through the plasma to the liver, where it undergoes further changes that prepare for its secretion into the bile. The amount of bilirubin produced and secreted into the bile is determined by the amount of hemoglobin destroyed. When the rate of red cells destruction exceeds the capacity of the liver to handle bilirubin, the yellow pigment accumulates in the blood cells, causing jaundice. Jaundice can also occur if the liver is diseased, like in general, or for example, hepatitis, or if the egress of bile is blocked, for instance, gallstone. The same question, those diseases which are related to different blood type groups